Welcome back to the OIS podcast. In this episode, Dr. Bala Ambati, the youngest person to ever graduate medical school and president and founder of iVeena Delivery Systems, discusses how to move from an initial concept to a final product that can impact patients. Let's listen to the wisdom Dr. Ambati shares.
Hello and welcome to the OIS podcast. I'm Dr. Paul Karpecki. I have the honor of getting to interview some of the top leaders and inventors and innovators when it comes to the category of ophthalmology and eye care and in entire space that's involved in. Today, I'm extremely excited because I have a chance to interview someone who's actually in the Guinness Book of World Records. You don't get to say that too often in a podcast, but also someone who is really making incredible strides in advancing for a treatment for a condition, keratoconus, that has limited options. Although we are able to treat at this point, these patients are some of the ones who suffer the most. So, Bala, I want to first of all welcome you to the podcast and thank you for making time in your busy schedule. As I see, you've just come out of surgery. Thank you for joining us.
Thank you very much, Paul. I'm honored and very happy to be here.
Thank you, Dr. Ambati. Tell me, well, got to start with obviously Guinness Book of World Records, the youngest, individual to have graduated from medical school. I think were you 17? I think at the time.
Yeah, I'm 43 now, so I have gray hair. I'm not as young as I used to be, but yes, I graduated when I was 17.
That is incredible. Tell me a little bit about. I'd even want to go back to kind of the upbringing of was this just part of the family culture? Were you a prodigy early? Was it other members of your family able to achieve that? What was the process in allowing you to achieve so much so young?
Everything I've done has really been due to God's grace and family support. And my parents and my brother were very instrumental in my academic upbringing. I learned a lot at home. I did a lot of independent study. Like all kids, I started first age 6. I went to public school. My education wound up doing 2 years of school every year. And that's how I was able to graduate from high school at 11. And then I went to NYU for college and I completed my bachelor's in 2 years. And so then I was off to medical school and I went to Mount Sinai and graduated in 95. Time is a gift. Time is precious, and the head start, so to speak, has given me time to meet a lot of great people, explore the field, really do deep dives in research, make mistakes, hopefully recover from mistakes, really build a good network of people around me who have helped carry these projects and other projects forward.
That is really impressive. And so it's true. You do 2 years in one. Now, you're going to have that. And it's good to make mistakes early. Failure is kind of one of those positives if you, can fail forward from them and learn. And a little more difficult to make to have something go wrong when it's late in your career or in your retirement or something. That's really a positive from that standpoint. And I love the family component that's played a role. So you, from Mount Sinai, then you went on to practice, I think, in Georgia, or you were on faculty there, and then now to where you're at in Eugene. Tell us a little bit of that path as well.
Oh, sure. So after medical school, I actually did residency in internal medicine and residency in ophthalmology. So I was double boarded in each. And then I did my cornea fellowship with Alan Carlson and Terry Kim at Duke. My first faculty position was at MCG in Augusta. I was director of cornea there for 5 years, and then I moved to Moran Eye Center in Utah, where I was for almost 9 years. My wife and I got married in 2015, and she had some family in the Northwest, and so we decided to move up here to Oregon in late 2016. So I joined Pacific Clear Vision Institute. It's a great private practice, 4 MDs, 2 optometrists, and very busy surgically with cataract, refractive, and cornea work. Bought the practice in July of 2019. So now I'm a business owner. It's been a very interesting first year and a half of business ownership.
That's for sure. With a lot of things going on during that first year. I commend you, though. You're obviously doing well through it. And I think on the backside of what we've gone through with COVID, things are looking up, and that's a good time now to be a business owner of your practice. Tell me a little bit about the pathway in research and how that developed. Was that early on in your career as well as part of what you did? Was that an interest that came later in? And how did it kind of lead even to the development of a drug like we're talking about in terms of with the company iVeena? But let's take the research path first.
Sure. I started doing cornea research in residency at Mass Pioneer. I learned from the best. I was one of the students of Tony Adamus, who went on to be a founder of iTech and then is now with Genentech. And so I got to meet some of the giants in corneal research in Boston, and continued doing research, throughout my academic career, where I had a laboratory at Georgia and in Utah, was NIH funded with 2, sometimes 3 R01 grants. And fortunately, I've been able to keep my lab team, and they've all moved to University of Oregon, which is in Eugene, and we're at the Knight campus for Accelerating Scientific Impact, which is an interdisciplinary laboratory research building that the U of O has established just a year ago. And so my core group of 4 scientists is up here in Eugene. We still maintain an NIH-funded laboratory. So it's been great to meld clinical work with research. When you take the patch off somebody, that smile of them seeing again is something special. After surgery, that's one of the best days of my week, is to help an individual person see again. When you discover something in the lab, you're having an impact on potentially dozens or hundreds or thousands of people in the future. And so that's also very important and very special. And so I think too often in medicine, we tend to say doctors should see patients and PhDs should do the science, but I think clinicians are uniquely positioned to appreciate what's important and what's feasible. And having clinician scientists be in the trenches with the laboratory scientists and lead and provide direction, I think is key to making truly transformative advances. As far as the keratoconus research that my company, iVeena, has been working on, this all started several years ago with an experiment in cells, in corneal cells. So you can take fibroblasts or corneal stromal cells from normal donor corneas, also keratoconic corneas, patients who underwent cornea transplants, and that tissue would normally be thrown away, but you can actually isolate their cells. And when you compare the two, what you find is something that's very interesting, that the cells from the keratoconic corneas don't express as much of an enzyme or have as much activity of an enzyme. It's called lysyl oxidase, or LOX. And so this LOX enzyme is deficient in corneal cells from keratoconic patients. And it's been published that LOX activity is reduced in cells removed from the epithelium of keratoconic patients who undergo crosslinking, and that it's reduced in cells from patients who underwent SMILE, and that lenticule of tissue, if you isolate it and compare normal SMILE patients to patients who subsequently developed keratoconus, the LOX activity is reduced there. This is not my work. This has been previously published by Dr. Shetty and various other people finding these LOX deficiencies. Separately, various other investigators have found LOX mutations in keratoconic families. So there's a large body of research that this native enzyme that's responsible for collagen cross-linking in the natural cornea is deficient in patients. And that contributes directly to weakened corneal biomechanics and corneal thinning and ectasia and so on that we all see clinically. So our hypothesis from a lab perspective was, how do we restore LOX activity? How do we restore the normal balance in the cornea? If you have insufficient LOX activity, the collagenases that are present in tears and corneal tissue, specifically something called MMP9, is unopposed. And so if you have insufficient LOX relative to baseline MMP9, that's bad for corneal homeostasis. So when we look into the biology of LOX, its requisite cofactor is copper. So my initial hypothesis was, can we add copper to these cells and restore LOX activity? And so that was the very first experiment we did. We took normal corneal cells, we took keratoconic fibroblasts, we added copper to both, and remarkably, both fibroblasts increased their LOX activity, but the keratoconic fibroblasts came up to the normal fibroblast levels. So we were able to restore LOX activity with just copper incubation in the keratoconic cells. So naturally that led to the next set of experiments in rabbits, where we demonstrated that copper eye drops could increase cross-linking. So that was proven by biochemical testing for looking for biomarkers of cross-linking, specifically something called LNL, or lysol-norlucine. Then we assessed corneal topography, and you could take a handheld topographer to a rabbit cornea, and we found that we could flatten the rabbit cornea by about 1.7 diopters with just about 6 weeks of these eye drops. And of course, safety. We observed no damage to the cornea, the lens, the retina. There was no copper accumulation in rabbit liver or kidney. There was no elevated plasma copper levels. So we did all of those safety experiments in rabbits as well. There was no injection or inflammation on the ocular surface. It was a very well-tolerated drop. So based on that cell culture work, based on the work of my team in rabbits, and then also getting a series of keratoconic corneas from Frank Price, where we showed that native cross-linking was reduced. I think we had a really solid foundation for moving forward with this strategy to restore LOX activity to try to increase native collagen cross-linking.
That's impressive. I mean, even the 1.7 diopters is kind of what we've seen in research with current, riboflavin affected corneal cross-linking with UV light. So that's impressive to see that in 6 weeks with a drop, a topical application too.
So that was in rabbits. We were very pleased to see that amount of flattening in rabbits. The rabbit cornea is about 350 microns. The rabbit cornea is normally about 50 diopters. So even though the rabbit doesn't have keratoconus, the rabbit cornea resembles mild keratoconus in terms of its thickness and topography. So it's a good animal model for this.
That's impressive. Now, how do you go from, let's say, a research idea? There'll be, listeners that are innovators that have come up with things, have looked at the research, may have come up with a possible theory of what's going to work, to, a company that you have like iVeena now, where you're, of course, serving as the chief medical officer, but having people surrounding you, creating board, getting to where you're at now. How does that process fall into place? I mean, who do you call? How do you start that? Is it just come to you with these ideas? How did you get to from that bench concept, which is quite impressive, and research and animal models to a great potential, to a company now, iVeena, starting, phase 3 trials?
Yeah, going from a project to a product is it's a long bridge, and you have to build a lot of pillars along the way, and as you say, get the right people together. So the company was a C corporation, established as a C corporation in 2015, and our initial investors included Park City Angel Network from Utah, Salt Lake City Angels, and a group of interested ophthalmologists, mainly cornea surgeons around the country. And so with the funding from the investors, as well as NIH grants, we're very thankful to NIH SBIR grants for the company. We've been able to move this technology forward. And 2019, we commenced a phase 1/2 study in Mexico to study the question of whether these eye drops can benefit human patients with keratoconus. So we started this trial with Codet Institute in Tijuana. Arturo Chayet was the PI in Codet Institute. Dr. Chayet's a very renowned cataract and cornea doctor, and he's done a lot of clinical studies from a lot of different companies. So as I recall, we enrolled over 30 patients, I think about 32 or 33 patients in our study, and 1/3 each to 3 different arms of the study. So a third of the patients who enrolled in the study received placebo eye drops, a third received iVeena 80, which is our proprietary formulation of these copper delivering eye drops. So a third received iVeena 80 for 6 weeks, and a third received iVeena 80 for 16 weeks. And then we followed everybody for 6 months. So the trial was structured to assess the impact of duration of therapy, as well as what happens after cessation of therapy. Probably took about 6-7 months to complete enrollment. We had the final visit last December, and so we had the summary, executive summary and clinical study report in January of this year. And what we found was remarkable. So first, there was not a single adverse event from iVeena 80. So that was very important in this phase 1 study, phase 1/2A study in humans, that there was not a single adverse event related to the drug. No change in cell count, no change in intraocular pressure, no injection or inflammation. It was very well tolerated. Second, efficacy-wise, we observed that the group of patients that received 6 weeks of therapy, they may have experienced a slight reduction in Kmax, maybe about 0.4 diopters of reduction in Kmax, but the group of patients that received the drug for 16 weeks, they had Kmax flattening of about a diopter relative to the placebo patients. And so we were very happy with that because we were seeing difference relative to placebo of a diopter at 4 months, whereas with surgical crosslinking, it actually takes about 6 months after surgery to get to that 1 diopter of flattening. And in addition to the Kmax flattening, which is what the FDA requires as the benchmark for approval, we observed reduction in corneal astigmatism on topography and some suggestions of improvement on corneal biomechanics. So outside the US, Oculus Corvus has biomechanic assessment capability, and Codet actually got the Oculus Corvus, and we were able to observe improved stress strain index on the patients who received iVeena 80 for 16 weeks relative to placebo. And that correlated nicely with what we saw in rabbits, that we had better corneal biomechanics in rabbits. So this kind of builds this complete picture of a drug that induces locks, cells in animals, increases the biomarkers of crosslinking, and in both animals and humans, flattens the cornea and improves the corneal biomechanics without any significant safety issues to date. So based on all of that, we have to go to the next step. We spoke with the FDA at our pre-IND meeting, and they laid out the path that was required. We don't have to repeat a phase 1/2A study in the US. We can proceed directly to phase 3 studies, talk through how many patients are needed and the length of the trial needs to be and all of these things. But we have a very clear path from the FDA as to what needs to be done to get approval. Along the way, the FDA also granted us orphan drug designation for this product, which is key from a business standpoint.
That's wonderful. I always have, felt for keratoconic patients simply because, when you finally could get them to a stable level, contact lenses, maybe early on or what's necessary, things would change again and they'd be all let down one more time. And then, even the technologies we had of the past, Intacs and things like that, they kind of worked for a bit and then they wouldn't. And then cross-linking, as you said, came along and it's kind of, halted things, but if you were fairly advanced, it halted it. So it didn't really necessarily reverse a lot. So this is a group, even when they eventually have to go to a transplant, the transplants had higher rejection rates. And so it was always a patient category that whenever I made the diagnosis, 10, 20 years ago, I always thought, it's going to be a tough path, but we're going to do everything we can together. It's exciting to see. And I love that there's an orphan designation for this, something that could really help a very needy population that would greatly benefit, and a topical application to that. And then for the future of iVeena, I think you're working even on the potential of progressive myopia. Now we're starting to talk about billions of people around the world. What's the theory or thought process and how that might work in slowing down that axial length progression by working on the cornea through this pathway, Bala?
Yeah, let me, if I can just finish up on the keratoconus piece for just a minute and then proceed to the myopia. Keratoconus is a condition that affects a lot of people It's and it causes a lot of burden on patients. I think somebody did an econometric model and found that the lifetime burden of keratoconus due to reduced productivity and wages and so on and educational opportunities is over 25,000. That's a significant burden for patients who are in high school or college or who are just out of college. And it's something, as you note, that is poorly served by our current stable of therapies. As you say, Intacs doesn't do everything you want it to do. Transplant has some significant risks, and contact lenses only go so far. And surgical cross-linking, as I think most optometrists and ophthalmologists are aware, it's been a bit of a mess. It's very pricey. It causes a lot of pain for several weeks. It has a significant healing time with visual fluctuation for months and a significant risk of haze and epithelial defect. Sure, cross-linking is way better than a transplant, but if you can treat these patients medically rather than with a surgery, I think everyone will be happier. I'm a surgeon. I love doing surgery, but I'd much rather do a cataract or a LASIK than do a cross-linking surgery. And I'm sure optometrists would much rather treat patients medically whenever possible. You asked about myopia, so I'll just pivot over to that. So we have demonstrated that we can flatten the cornea. I flatten the cornea every week to treat myopia with LASIK. Right. So, if we can flatten the cornea with a pharmacologic eye drop, then that can have a significant impact for progressive pediatric myopia, which, as you noted, is an epidemic, and that's not treatable by LASIK, right? You're not going to operate on 8-year-olds with LASIK. And so what we have found in our preclinical work with a related product that also delivers copper to the eye, it's called iVeena 85, is that it can deliver drug both to the cornea and the sclera. So we've formulated it such that it increases not just corneal lysyl oxidase, but it increases scleral lysyl oxidase. So we've observed increased scleral cross-linking in guinea pigs, which are a model of spontaneous myopia. It's a spontaneous animal myopia model. We've observed reduction of axial elongation in the guinea pig eye, reduction of the vitreous chamber depth in the guinea pig eye, and a hyperopic shift on cycloplegic refraction done by a masked pediatric ophthalmologist of the guinea pig eye. So in animals, this iVeena 85, which is the product name for the myopia product, has looked very promising. There's been no safety events where seeing about 1.5 or 2 diopters of hyperopic shift on cyclo, and a substantive reduction of axial elongation rate and vitreous chamber depth. So we have some more experiments to do with animals. We're not just guinea pigs, but also rabbits. But we are gearing up to do a phase 1/2A study of pediatric myopia, and that hopefully that'll be a longer observation period, a year. So we won't have final data on that till late 2022 or early 2023. But I think it would be a huge market for iVeena.
Very exciting. What would be your advice, as kind of a final question here, Bala, for some, a clinician who, or a surgeon or an optometrist or ophthalmologist who finds themselves, interested in research, may even have come across a concept that could make a potential impact on patients' lives and eyes. What would be your advice for them moving forward, and what steps they might take in that direction?
So the journey from idea to impact is a long one that requires patience and perseverance. I think it's key initially to secure the IP with a good patent attorney and put a team together around you, develop the business and the product. So, figure out what you need to raise money for and what to spend that money on in terms of cell culture work or animal work or technology development, and then raise enough money to get you through some reasonable number of patients in a phase 1/2A study. So if you talk with your friends, who tend to be other ophthalmologists probably, or optometrists, and you can show that there's leaders in your field willing to back you, that makes it easier for the professional investors, such as angel networks and so on, to view you and your company as an investable concern. To build the enterprise, you need both the confidence of subject matter experts as well as the confidence of professional investors. And so I think that's what's made iVeena successful to date is the backing of both angels and ophthalmologists, as well as our grants.
That is fantastic. Really wonderful insights. Thank you for your openness and sharing kind of your history, and you're so humble. And in terms of that, I've been in the Guinness Book of World Records, but also the accomplishments and what you've done and where you've taken it. And congratulations on your path, and now with your wife and your family in Oregon, but also even the insights for someone who may have a passion for this, have come up with an idea that's innovative but doesn't know the next steps, and sharing what you've done and your continued success. And I expect it'll only continue. Just in the brief time of getting to speak with you, you could see there's so much potential here and so much good leadership you and the team are providing. So I want to thank you for taking time out of your busy schedule to share those insights with so many listeners in the eye care field. And certainly, my hope is that the continued success, iVeena will do well. It's going to serve an incredible need. And it's great to have someone of your caliber and ability kind of carrying that forward.
Thank you so much, Paul. I appreciate the opportunity, and my warm wishes to the whole OIS community. Thank you very much.
Thank you.
Thank you for listening. We hope the great information Dr. Ambati shared will be useful to you in your pursuits. For more on iVeena and other companies addressing myopia, register for the upcoming OIS Myopia Innovation Showcase, taking place on April 29th. And come back next week for more insights and information from the OIS podcast.